So our topic today is the bio (333) comparative animal physiology. Okay, I want to stress here physics, right? What is physics? Physics, have you done? This is biology class, right? Or a physics class. Don't ask me about physics, right? But the physics is of living organism. So here the living organisms, in the case here, plant and then animals. Okay, and then animals that consists of in the human as well, derived from that one. And then you can also get the microbes, microorganisms. Okay. So everything is going to be the physics. Physics is something like a function, the work, right? Work of plant, work of these animals, and then human microbes. What this work they are doing it, but what functions inside this organism to live. If the animal is living, means how do you say it is a living? The chair is there, the table there. Are they living? Why? Huh? No blood flow. Okay, that's good. No metabolism, right? Okay. So if a living organism, a living life, okay, if a life is there, that means there is some process it should be done, some work should be done, correct? So how those work is being carried out? So that's what we are going to discuss about. Okay. I put it like organisms, okay. Now that's we call the physiology. What is physiology? We are giving the definition. Okay. Mainly the physiology means how things, as the living things, work. Okay. And physiology, a part of physiology is also in the development of an organism. Okay. Development, what do you mean by development? How the living things form. Okay. So it is developed and then it will do the physiology. So physiology of development and development after development, the physiology, how things are working. Everything put together. Because the physiology means how things work. That you should understand. That's what the first point you should remember in this one. And then development is how things form. Okay. Now, how things work. Now let's take that point. How things work. Suppose if this is the organism, if it doesn't work, as you mentioned before, it needs energy, correct? Right? So the energy should be derived from where? Food. Okay. Suppose this is an organism, is around one. Okay. So it consumes food. Okay, check food. And then the food alone cannot give this energy, but it should be metabolized. So it should take oxygen from the environment, outside air. And then it will expel CO2, carbon dioxide. When the food is digested and everything is taking place here, and that also produce some waste products. So the waste product should come out of this organism. Okay. What waste products here? We have ammonia, toxin, and this ammonia become in the form of urea. Also water, excess water and excess salt which will come out of this organism, any organism. When the food is coming and that is digested and then oxygen is there and then waste product should be expelled out. When you do this, you need a circulation, then only it can be done. So you have a circulatory system. Circulation of oxygen, circulation of carbon dioxide, and circulation of waste products, and circulation of the food when it is digested in the food. And then only it can be done. So when you think of physiology, you study about how things work. If a work should be performed, you need energy. How do you get that energy? If you don't eat breakfast or lunch and dinner in the night, what will happen to you? You will say, I'm exhausted. I cannot do any work. Don't come to me. Don't talk to me. I am tired. Is that right? Meaning you need some work. If you want to perform work, you need energy. That energy is coming from the food. So if the food is getting in, and it's not only the food. If you give the food and keep quiet, no. If the person is dead, can you put a funnel on the mouth and then put the milk or anything into it? Can he? Just circulatory system is not working, and the respiration is out. So no oxygen, it cannot do it. So the living organism that requires all this process in one package. But in our convenience, we are going to divide into part and parcel of small bit and pieces of this work, and we are going to study in this class. Right? Does it make it simple? Okay. Now, all this process, consumption of food, oxygen and waste, pushing out the waste product and circulation, how they are regulated now? Regulation. How they are regulated? Number one, they are regulated by hormones. Number two, they are also regulated by nervous system. And this nervous system also include behavior. For our purpose, we are not going to discuss about the nervous system or behavior in this class, because the animal behavior, which is being separate course with Dr. Vineman, is teaching on that part. So he will take excessively of the nervous system as well as the behavior aspect. Okay, but our part is here about the hormone regulation of the physiology. Okay. Any question? Okay. Next part, we are going to study the function and work of the organism in a different level. Okay. For example, we can study this organism. Okay, we are studying organism. What level we can study? It's the cellular level and tissue level. Okay, that cellular level. We can study the work of a cell. Okay, if you want to study an organism, we have to study a particular type of cells. Okay, for example, if we want to study the digestive system, how the animals are consuming food and digested and then excreted or the waste products. Okay, to start with the digestion, we need to have what cellular process which is in the digestive system and the functions and everything. Just one example, like the circulatory system, blood and blood cells, right, and kidney cells for the excretory products, and the lungs and the tissues in the lungs that involves for the oxygen consumption and the expulsion of carbon dioxide. So, okay, cellular level. I'm giving an overall picture and then we will go into the depth. Okay, and then we are going to study the molecular level. Molecular level, meaning how the cell is processing in terms of the molecules of food molecules, or the oxygen molecules, or carbon dioxide, or urea molecules. So everything is going to bit and pieces, how the ion is conducted from one to the other. Okay, so on a molecular level. So this part is, we call it as physiology, correct? How the animals work. This part is a physiology, okay? Now, we are going to see on the how this physiological process which is present in the environment. Suppose it is in a fish. And fish and the environment is a cold water, and all on a sudden is a warm water, and all on a sudden is salt water, from fresh water, salt water. One environment into the another environment. Okay. So when you compare the physiological process to the environment, okay, when you compare the physiology or adapting to the environment, adaptation part. So that is, we call it as a comparative aspect of physiology. So in total, what you get here in this organism, comparative physiology. And the physiology to the comparison to the environment, here we call it adaptation. Adaptation to the process. How the physiology of this organism is being adapted to the environment. When you compare the normal process and how this organism is adapted itself to the environment, that is called, we call it, that aspect we call it is comparative physiology. This is the normal physiology, and when you adapt to the environment and different condition, and that part is the comparative aspect of the physiology. Do you follow this part? Yes? Okay. If you have one life, if you have one organism, okay, living in terrestrial, in soil, okay, and their digestive functions and everything is the same. All the soil, terrestrial animals. Suppose if the same animal is getting into the water level, or if you're getting into the another environment, the soil is the one environment and this water is the aquatic, another environment. If it changes, so how this digestive system from the terrestrial environment that's being changed to the environment, the new environment of water, so the animal is different. Okay, so in that environment, how they adapt itself. So that part is a different species of the animals. So in the aspect which I mentioned before, the evolutionary aspect from one organism into the another organism, and the same function, how they differ due to the environment, due to the adaptation, due to the evolution of the different species and different organs. So this aspect, again, we call it as the comparative physiology. This is the definition of this class, comparative animal physiology. So when you do the environment, now I'm going to from there. So what we get it in the environment, many organisms. Okay, and the many organisms, they have many environment. Many environments. For example, in the air, water, as well as inside the soil or inside one animal's body, or so the many organisms, they can live in many different environments, like a hot and then cold environment, hot environment, high salt intake and low salt, and fresh water and salt water, right? Different one. So that leads to many life histories. Many organisms, many environments, that leads to many life histories for this. How they do the adaptive modification. Adaptive modifications. Okay. Example, fish live in water. So when you take a fish into the outside the water, what will happen? It will die. Make a frying pan, fry and then eat. Right? So that is very simple. So if you have a bird, Bat can live outside, that's not in the water, but in the air. But it will go into the water, dive into the water, and pick up a fish. And then that's this feet, right? So that's the part. So this environment is different for this, and this is for this. It's essentially different. So there is some adaptive modification for the fish as well as the bird. Do you follow? So as I mentioned, many organisms and many environments and many life histories. So the life history of fish is different from life histories of bird. So we are going to deal with this. So I am giving an introduction to the entire course today. So that's why I want to know where we are and how, what is the importance of this class and goals and objectives, everything. So our objective to study, compare from one organism to the another organism due to the changes in the environment and also the different life histories and the adaptations and everything put together. Okay. Next one. How many species are there now? How many species? How many adaptations? So there are varieties, right? So if you think like 1.5 million species, this animal kingdom, and that generally estimate with around the beetles alone, beetles, that's a maximum of 30 million different types. Meaning you have 30 million different species of the beetle alone, they have 30 million of adaptation. So different species means there are different adaptation which is coming up. Okay. See, the adaptation, one of the human, species which we talk about here, and it depends upon the environment. The Eskimos, they live in Eskimos in the north of Alaska, and their behavior and their digestive, their systems and their physiology is a bit different from Texas, where the more sunshine state here. So they adapt with each other. You follow that? So different species, even the same species and the different temperature, yes, we have the adaptation. If you have different species, yes, certainly there are different adaptations. So when you think of the different species in an evolutionary perspective, have you ever heard of the phylogeny? Phylogeny, the classification according to the evolutionary perspectives from, all organisms are derived from common ancestors. That's one of the evolutionary theory, right? All organisms derived from a common ancestor. So this one is like an evolution. I am not going to argue with the evolution or creation, but I find one commonality on that part. Yes. In the creation part, God made everything on man on his own life. That's one theory. The another part of the religion said God himself is divided into different species. Okay? So when you think in all put together in one part as a life, if there is a life, and that should be a common ancestor for the life, that you can call it as a common ancestor, or they call it as a religiously speaking is a God. You can put it that way. So either evolution theory or creation theory both have one common ground. Okay, they said common ancestor. They won't call it as a God or anything. But here religious people, they name that common ancestor as a God, and then they talk about it. Because both of them in this case are life. If there is no life, that part we are not going to deal because it's a life science. But it is physics and chemistry, it's particles and atoms and molecules. So those of inanimate, the lifeless, there's no lifeless molecules, that constitute for the life. The life needs the other atoms and molecules and everything put together, right? So it's a bit of a hierarchical meaning. The life process needs lifeless molecules and other chemicals and organic chemistry and then inorganic chemical and that particular chemistry. To function put together, we need physics, and that's what the physiology is all about. Okay. How many groups on phylogeny? Just for the fun of the classification, you can have three groups like bacteria. Okay. You have another one, archaea, AEA, and then eukarya. Eukarya. Okay. There are three parts. This one, and they also have, yeah, these eukaryotes, which is having a nucleus, and bacteria no nucleus. In the structure, the bacteria is there, and this is your flagella, and then you have a naked chromosome or the DNA only there, and there is no nucleus. But whereas the eukaryotes, you have the nucleus and mitochondria, and everything. This is the nucleus. This is the envelope. This is the envelope. Okay. So there's a differentiation from one organism into the other. To establish these functions, okay, there's a lot of complexity. Which involved from bacteria unto the human, from bacteria to the eukaryote, prokaryote to eukaryote. So there are certain things from one single cell into a multicellular organism. There are a lot of complexities involved. But one common aspect is bacteria also feed, eukaryotes, the higher form, also feed. It also produces energy. Bacteria also work, and the eukaryote, they also work. They do all biological function, they perform. Do you follow? They also perform. But it may vary, it may differ. Why? Different environment. And then the different species, that's what we are going to discuss about, the comparative aspect of physiology in a different animal species. Okay? Any questions up to this? No? The pattern of same pattern in animals. That's what we discussed. Just now I told you about it. Same patterns in animals. Okay. For example, sponges, jellyfish. Okay. And then you also have insects, snails, starfish, human. Okay. The development from the bacteria to this, you have got in a evolutionary perspectives of 1 billion years, billions of years to the modification. Okay. And the physiology is the common. Physiology of the function. That meaning the cell work, how the cell work. Okay. And then how the cell communicate with each other. And then cell membrane. So all these different species, they have the same pattern in animals when they do the digestive system, and the pattern is, follow the same pattern, consume, as I mentioned before, energy, feeding, and then the transport and the work and everything is the same one. The physiology, how the cell work, everything is the same. But the animals may vary because the environment is vary, their feeding habit is vary. So that part is only the variation. So we are going to study the variables on one side, and other side is the commonality or the pattern of that. So we will study one by one, okay, system by system in a different aspects. Okay. now we'll go into the general physiology. Okay? General physiology, meaning I'm going to talk about just physiology. I'm not comparing it here, just one aspect, general physiology. Okay. For example, in a bacteria and then human. Okay. Do that. This is the organism where it can consume oxygen and the carbon dioxide will be produced. That is going to be expelled out, and then the food sources like food in the form of energy. See, the energy is stored in the form of food. For example, if you take glucose, okay, high content syrup, and if you drink more of energetic drinks, see, that will produce of energy into your system. There are some drinks without any energy value or calorie value, so that is a pure of chemical that gives good taste for your tongue, but it won't give any energy. For example, Splenda, right? Do you know Splenda? There's a dietary sugar, right? Which is a dietary people they eat. That's a sucrose, the sucralose. It's a sucrose molecule that has one hydrogen, and they put one chlorine molecule is there. So when you put that, you get 200% sugar. You have taste of that sugarish. So that compound is not metabolized in your system, in your body. There is no digestive system. Not even a bacteria won't digest the sucralose. So whatever, if you consume that Splenda, which will digest, and that is going to be eliminated either in urine or in the feces. It will come out. It is not going to stay inside our body as well. So it is a normal inert molecule. Okay, so that is energy less, but you have got, as a taste, that's different issue. In this case, food means you should give some energy. So all the energy in the form of bond which is present in the food. Whenever I saw food, I could imagine it is a chemical bonds. The different atom, different molecules. That's what we are consuming every day, every now and then. So those food is going to be processed in our system, digested, and then it is being absorbed. And inside the cell, that eliminate that energy bond. The bond is releases the energy. That's what we need to work and talk, listen, and whatever they perform the work. Everything's derived from that bond energy, which is derived from the food. So that's why I said, if I say food, means here this energy, we are consuming the energy, and that is going to be present in our system, and it is being digested and using the metabolized in presence of oxygen. And as I mentioned before, the waste products like ammonia and other toxin after the digestion. But all this process will take place if there's a proper circulatory system should be established. Circulation. Is a general physiology I'm talking about, okay? And this is further being regulated, as I mentioned before, regulated by hormones, okay? And then also cell-to-cell communications, cell communication. And then the development of this organism, or the reproduction, development, all put together, it is called general physiology. This is a one organism, it's a general physiology. And it is not as a comparative physiology. This is a general physiology, which we have done on an early stage. Now, what is comparative physiology now, which we discussed now? Comparative. Comparative physiology is how one organism is different from the organism 2, and 2 is how is different from 3, and 3 is different from 4. Why we need to study all this? Number one, we need the investigation. investigation perspective and scientific curiosity. Scientific curiosity. Hey, I want to know how this bird is flying. I cannot fly. One example, a curiosity. Fish can swim, which I cannot swim now. I have to do practice for the swimming lesson, then only I can go onto the sea or the pond to swim. Okay. So how a snake can swallow a frog, which I cannot swallow a frog by myself. How? It differs from one organism feeding habit into the another organism's feeding habit. It may differ. So it is a scientific curiosity. I want to learn. I want to do something. So when you do scientific curiosity, you have to spend thousands and thousands of dollars to learn, to do some experiments and to some results. Then you will know why the birds are flying, which I cannot fly. Correct? Do you follow that? So the comparative physiology is the knowledge which you need to know about the scientific curiosity, a knowledge to be learned. For that, to learning the knowledge, you have to spend money. So to learn, to get a degree, you need a knowledge. To get a knowledge, you have to spend your tuition fees. Correct? You spend money. So for each and everything onto the study of the knowledge, you have to spend money. So here the scientific curiosity or the investigation of the different process that involves money. And that also involved in the economy. Okay. So we need to publish some of the books and the research. So all backing the comparative physiology. So to study any single aspect or to study a simple process, digestive system or any other process, okay, we need a model organism. What is an ideal organism? Model organism to study a particular process. In olden days, the scientist Krog, or Kroger, Alexander Krog, and then he has argued, given diversity, okay, that many problems, because of the diversity, different organisms, right? Diversity. Want to study many problems in this one. And then if you want to study one problem, that's a single process, we need a model organism to study, to explain, to understand your kidney function or the digestive system or circulatory system. Call it as an ideal organism. Ideal organism. So they have developed an ideal organism to study a particular process, a particular system or organism. For example, have you ever heard of pig? And the anatomy of pig that provides the ideal nervous system. It's a primitive nervous system, but we can easily understand. I'll explain in a different way. If you want to study a nervous system of a human being, dissect into neurons and getting into the spinal cord into the brain, and brain is full of neurons and every complexity is there. What is the motor neuron and then sensory neuron and different types? You are lost when you get into the human beings. Instead, if you have only one neuron and it will do one function, very simple, and you can study at the different sensory input and then output very clearly without any complication, any complex issues. So to single out a function, you have to have some simple organism and simple method and then simple way to understand. In a scientific curiosity, whatever we are dealing with that, we want to learn something. If you want to learn very complex process, we have to go on to the basic. We have to go on an elemental level. If you want to become a voracious reader or the speaker or orator, you have to know first of all what? An alphabet, A, B, C, up to 26 letter. What is A, B, C? You should know the alphabet. Without that, you cannot go or construct a word or write and everything, right? So, in the scientifically speaking, if you study the complex organisms or organs or the systems or the function, you have to go an alphabet of that particular organism, of this function. This alphabet, what we are talking about, if you have a one simple or single organism and their function, if you understand, then you can correlate into the multicellular, multiple level of organization of this other organism, which we can study. So, to study a simple process, a single process, on one particular system function, we can go onto the different system. That's what the squid is an elementary aspect of neuroanatomy. Elemental. That's a very simple formation of neuron or nervous system. So when you study this, then you can compare this particular function into the higher or the other organism. Do you follow that part? Why we have to study onto the lower part of this animal kingdom, like a squid. So the knowledge empower. To get that knowledge, you have to go very simple. Understand a complex problem. Complex problem. The neurons, that's one of the primitive one here. So, what the scientist Krog, and he suggested ideal organisms for any problem, or any system, any problem. Think of, we have to go analyze different animals and then finally find out, hey, this is the one ideal organism, which we can study elaborately about the nervous system. These organisms create. Like that, there are different systems, different organisms are there. Okay. In other words, they are called animal modelless. We know about the classical models on our dressings and makeup and other aspects of models. Like when you go on the TV ad, when you switch on, you have a different model, ad models. They are coming, they are showing their hair or skin, whatever they want to display. They want to display about the coat, or they want to dress. Yes. Sorry. Okay. Here now, I pulled the mic. Harry, what shall I do? Volume, can I reduce the volume? Put it here now. So static. Still, you are getting it or not? Go ahead and then let me finish this part. And at what stage you are not able to hear properly? Okay, great. Okay, now I pull the mic in near to me so that you can clearly here now. Is that okay? Now, okay. All right, Yang, there is a problem. Static. I can hear you clearly. Okay now? Oh, is it problem signal line? Oh, okay. I think she will call someone. Yes, that's what I'm thinking that one. Just I want to give you a break for 10 minutes and let them solve this problem and then come back. Is it okay? Great. All right, great. I was getting onto the squid anatomy, all right? That's the previously. That provides the nervous system and that give a simple animal system into the complex, organs. So that simplify, we are choosing different animals, a simple animal, right, to study a complex mechanism. So that's a part which we need to do animal models. So I want to discuss about it. Okay. So the animal models serve as a model organism. So in this model organism, you can study different functions of a complex system. Suppose, for one example I'll give you. I want to study a drug, okay, whether this drug is inhibiting a cell division. For example, you want to study a cancer. You want to study that. So the drug, instead of taking that drug directly onto the human beings, if I take this drug and then put into animal cell culture, a simple and a single cells, and then check whether the cell division is being arrested or stopped dividing it. Okay, so cell division, inhibition of cell division, that's a very simple process. One cell become two, two become four, four become eight. So at what stage this drug will inhibit and what process that will take place that to inhibit. So that is one of the model. So this is a cell line as a cancer cell model, right? Model organism, right? Like this, if you want to study, you can also study the same principle of different drugs onto mice and Drosophila, okay, and functions of one organism, right, can be applied to another organism. That's one other principle. Function of one organism can be applied to another organism. What? These are model organism. We call it as animal models. What is the purpose of this animal model? Why do we study this animal model? Okay. The function of one organism can be applied to another organism. So this will help us to reduce the cost, to understand the toxicity, to screen several drugs in a short period of time. All those be useful. Which company? Pharmaceutical company, pharmaceuticals, as well as the beauty shops where the different type of creams and other things, and then hair. So all those industries, the cosmetic industries, I'll put it in that way, as well as the medical industries um, for to study a particular type of a new type of surgery, they want to perform in the animal. They want to perform in pig. They want to perform in monkeys. They want to perform in a new model. They call it as a human beings and a prisoner of war. They act as a model organism for the patients, real patients. They want to test. After testing onto the animals, they want to test in the human beings. Who is the guinea pig for that new novel drug or new type of surgery? The prisoner of war. That's what they did. At that time, there's no human ethics. So they did First World War, Second World War, they did all those things. Okay, and we are dealing with this in our bioterrorism class too, on this aspect of modeling this organism to the humanity. But that is the worst part. But we are not going to cover that. But here in this one, we are going to deal with some of the model organisms. Okay, we'll study. So what we need to study these animal models, what we need excess of diversity in these animals, right? Diversity in order to use it for the animal, diversity in animal model. So we have to know the different animal model to check it whether that process is different or same pattern which is there. You want to check. So that's why this class of comparative animal physiology is all about. If you want to do that, if you want to perform this, you need what? To money, economy, and then knowledge, as well as you want to study, publish the work on a research paper, a research article. You have to publish the work, what you have found. Yes, I found this particular organism is good for studying a digestive tract, a particular type of malignancy, and this is a very good model organism. In some case, they create the model organism. They introduce a gene of interest to that organism. They call it transgenic animals. Suppose I want to study a particular drug and it will interact with a particular gene. So instead of using that drug on the human being, I can take that gene and put it in the animal. And then the animal became a transgenic animal. And then I feed to this drug and then check whether that is going to be inhibited or not. So one drug which I did my own experiment, I'm doing it, now I'm doing it with the limonene, with the orange fruits. Of course, it is a hormone dependent cancer cell, and breast cancer cells, it kills the cancer cell. But hormone independent cancer cell, it won't kill that cancer cell. I want to know why it is. So my major input is a problem, a model organism. I took that cancer cells and the human cancer cell and put it into the mice and then grow inside the mice. The mice is growing the human cancer cells in that xenotransplant. And then I inject this drug onto it, whether it inhibit or not. It didn't inhibit. So, but it inhibit along with the regular chemotherapy. So it is not inhibiting on its own, but it helps to inhibit along with the regular standard chemotherapy. So in that way, yes, it is a good thing. Then I have to publish the work. I have to write a project and then have to publish it. So, and this fund is being funded by USDA. Okay, then citrus farmers, and they want to give this money, whether we can supplement orange fruit juice to the cancer patient along with the chemotherapy. If you drink orange juice, there is no, if you drink orange juice and then if you cure cancer, then there's no cancer hospital. People won't suffer from cancer. They drink orange juice, they're happy. But it is not the case. So it is not going to help anyway. But at the same time, it is not inhibiting the chemotherapy. Sometimes what happens, some of the food item, what we take it, that inhibit the regular chemotherapy. That also it involves, because the food which cancel the chemotherapeutic effect. So how do you study? The same way, study the cell culture model and the animal model, and then will work. So I've done several work on inflammation, arthritis, and then cancer. So all in animal model I have done it. So that's what I thought. Okay, this is appropriate. My subject, compared animal physiology, I can go through it. Okay. So one of the site which I would like you to go onto the reserve, electronic reserve, which I will walk you through at the end of this lecture too. Okay. So diversity in the animal model, in order to use it. So we need a different type of animal for different purpose. Okay. So how many model organisms are there in the outside? How many? How many model organisms are there? The National Science Foundation, NSF, they have got different perspectives. They have got like a bacteria, ideal organism for bacteria. They can work with 5 different bacteria. And protista is 4. Fungi, model organism, 4. And plant organism is 9. And the animals, how many animals in the model organisms in the animal? 65. And this number is growing more and more in the year come. It is growing. Okay. These are the model organism which is currently available to check, to use it. Okay. So what we discussed so far is about our scientific curiosity. Okay. And now I want to go in the PowerPoint slide, and then we will go back again. Okay. I don't know whether you can clearly see from the other side. Could you see from there? Synco Ranch. This is a table on the from all these PowerPoints are derived from your textbook. So if you have the textbook, you can check the table 1.11. Common physiological processes and the chemical and physical principle that relate to them. Here the Ohm's law, Boyle's law, ideal gas law, and gravity, and inertia, momentum, velocity, drag, kinetic, potential energy, laws of thermodynamics. All are physics. That's why I told you about physics involved in the physiology process. Gravity, here you got the blood flow, animal locomotion, and gas exchange in lungs and gills as the Boyle's law, ideal gas law, which is applicable to it. Okay, muscle contraction is kinetic and potential energy. Okay. Anabolic and catabolic metabolism, that's the laws of thermodynamics. These are the chemical and physical principles which involved here. So we will discuss that, the whole. And here you can see this organism level. I'll just put the organism level, the whole organism, how they behave. If you pick up that particular joint of the organ level system, where the muscle alone, look at the skeletal muscle and the histology of the muscle, again the organ tissue level, or and then on to the cellular level, where the myofibrils. So fibril constitute a tissue. These are the muscle cells constitute a tissue, muscular tissue. And the tissue and bone form the organ level here, the skeletal muscle, and then they got the organ system level, how they function, and then this function relate to the organism level. Look at this one, cellular level, skeletal muscle cell, the macromolecular level are the sliding filament, and then the myosin as the molecule, and the actin is a filament, and then how they constitute and to slide each other, one protein to the another, the muscular contraction. So these are one simple example. We are looking into that muscle contraction, and we are at the molecular level or macromolecular level. Okay. And some of these are the large external fluctuation of this temperature, and then how the small internal fluctuation in fish which occur. So it adapts the excess of outside temperature onto the inside. It can adapt. So that adaptation process as a physiology we can study. And these are some of the physical principles which involved in the thermostat. Could you see that one, cold and hot with the spiral? So when it is hot, it will move out. So this connection is over. There's no current is passing through and heating this coil, heating coil. Suppose if this become cold, then it will come and contact with the electrical current, and thereby the heating coil, the bimetal coil is be heated up, it will become hot or cold. It's a thermostat. Okay, that's the mechanism. So that's the same thermostat mechanism existing inside the fish to adjust the temperature. So that's the process, the physiological process we want to study. And this is a conformer and regulator. Then how these are the physics. I mean, how that's been regulated. Here, the zone of stability where homeostasis is maintained, and this level to this level where the fish can maintain the homeostasis. If it is excess, its temperature or it may go up or it may come down. So, and ultimately, it will maintain this one by the regulator. Okay. Conformer again. I mean, this is the mechanism of how these regulated by the feedback mechanism. By the physical principles which is involved. And also these scientific inquiry and scientific results have been published in different journals. Look at this list of journals in your textbook also. You will find Journal of General Physiology, American Journal of Physiology and Pharmacology, European Journal of Physiology, Comparative Physiology and Biochemistry, Journal of Comparative Physiology. So all you can find out from these journals. They are publishing their work. Again, the different journals. And this is how this has been abbreviated. Scientific inquiry. This is the chapter 1. This is the basic principle what we have done so far. And then the chapter 2 is again the structure of there's a spinal cord and a different experiments and then how they do perform this experimental different type of experiments and scientific inquiry using the animal model. These are one of the mice and animal model to study the spleen producing the antibody antigen, fuse the cells, and these are myeloma producing a monoclonal antibody. And I suppose we discussed elaborately in the immunology class as well. And this is a one of the model where they can produce an antibody for production. Okay. These are the neurons and how the neurons are excited with fluorescent labels. Okay. And certain techniques which are involved over here. Okay. Where we can study the immunohistochemistry and other techniques and the ELISA assay. So this one is a plasmid, how they genetically engineered on a bacterial chromosome. All physiological process, they need experiments, and these are the experimental method. Okay, and I'm going through this because this is I'm not going to explain individual experiments because there's a full of cell biology, and this is on our histopathology or histology. What we are doing it in our lab on the laboratory where we can prepare the microscope slides, and this is about the microscope. Some of the techniques or technology which involved to study the physiology process. Okay, it's the confocal microscopy, but this is a normal onto microscopy. And then if you have a the same one the previous slide, but now we can have a clarity by how the tubulin and the one cell become two cells, cell division. Now we can see these retina of the eye in a confocal microscopy and in a different perspective. So different kind of microscopy we can study. Okay, and then bright field, phase contrast, and then these are different field in the microscopy. And these light microscopy, electron microscopy changes and technology which is involved. So I'm just these are some chromatography and all of them are the biochemistry which you will study in a biochemistry lab and the principles which are involved. So the physiology is full of technology. Physics, how to study, which I mentioned before. If you want to study a function, you have to go back and then do some experiments and publish the results. And these are the methods which I mentioned before. In the chapter 2 is exclusively of different methods or experimental protocol and procedures which has been available. The lab, wherever you do physiology or biotechnology lab, cell biology lab, histology lab, or immunology lab, whatever you are doing it, the same principles which is there. So that way, physiology can be studied by your practical method. Okay. Now I have another one of chapter 3, but this one is having an audio component which is in it. Okay.
Molecules, energy, and biosynthesis. First, we look
So
into
I'm not going
the
to
power part of the
details of that. So you can go through that PowerPoint by yourself, because the basic principle which involved on the molecules and energy and everything put together on the chapter 3, you can read it by yourself. I'm not going to assess any exam questions on those chapter 3. But according to this chapter 1 and 2, yes, I'm going to assess you from that. Okay. So what my point is to check the questions on the chapter 1 and 2. Okay. So what you are going to do, you can read my PowerPoints, not PowerPoint, the class notes, which I just now I've given to you, and perhaps you can have a homework on that. I'll give you, and chapter 1, okay, it's not a chapter 2, is not, just chapter 1. There's some questions over there in your textbook, okay. You have the summary on the page 14, okay. And then you have a review question. You read those summary, and the review questions 1, 2, 3, okay, 4, 5. There are 5 questions over there. So I need to focus on review question 1 and 2. Okay. You could just study, summary, and then review question 1 and 2. That's your homework for that. Next week, I'll give you a 1 week time for that. You just study, and you can either bring in a hard copy, or you can send me an email attachment onto the Blackboard. You can send to me. Okay, so that part. So this is an exploring the physiology, and now I would like you to study about the next topic, which I want to discuss about the digestion part. Okay, the chapter 15 in your textbook. Let me see that I have it here. Yeah, these are some of the PowerPoint from your textbook. Okay, it's a different view, but I'm going to give you my lecture part first, and then we will go through this later. Okay. Any questions now for the part of chapter 1 and 2? From Synchronch. I could see some more students over there. I know that 4 students, and can I get your name, please? Because I want to get an attendance. How many of you are there in Synchronch?
There are 7 students here.
Oh, okay. What's your name? Ellen Hawker. Yeah. Next one. On your right side. Yeah. What's that? Gloria? Gloria, okay.
Lillian Garcia.
Lillian Garcia. Oh, okay, okay. I couldn't hear you. Okay, I'm sorry. Okay. Next student?
My name is
Shaji Han. Okay. And then another student? Amma? Amar. Okay. Okay. Good. Good to hear from you. So, could you follow this class now? Some sinker wrench. Do you understand? Without any static? Okay, good. Sugar lamp. Okay? Yes, go ahead.
If we don't have a book yet,
Okay.
how can we submit?
Okay, for those who have not received the book or they don't have the book, why don't you write the summary of what we have in the class? Write the summary of the class notes and submit. Could you follow? Yes. Either you can do it with the book, or if you don't have a book, you have the option to write the summary of the class notes. Okay, within 2 pages is enough, 1 to 2 pages. It's not, I'm not asking everything, every word, but what you understand from the feedback. Yes, there is.
Do we send it online or do we?
Online, yeah, online Blackboard, yeah. WebCT have email attachment to this, so it's easy for you as well as me to do that. Okay, all right. So now I would like to go back onto the document camera. Okay, now, okay. All right. I want to, yes, go ahead.
Starting a new chapter there?
Yes,
Chapter
chapter 15 in your textbook. Okay. But it's not everything onto the book, but I want you to onto the digestion part. Chapter 15, digestion. According to the syllabus as well, okay? Okay, if there is an organism, as I mentioned before, if you want to study a digestive system, you have a model organism, right? So the model organisms, what if you mean to digestion means feeding? I'm getting hungry, right? It's getting lunchtime, right? So with a feed, you are going to feed. So without mouth, how you are going to feed? See? Yeah. You have a mouth. To food should go in. And this body will go there, right? And what happens here, in some organism, there's only one orifice, okay? Orifice, meaning one hole there, where the food can go in, and it will go in, and then it will digest, and then some of them is absorbed. And then there is no anus, but it will come out the same way. Okay? So do you understand? Just like a box. Like a box or a bottle-like thing, right? So everything is put it in, and it will go, digest, and everything is absorbed, and then it will come out, because there is no other way to come out. So the food is going one, and then the waste is coming in the same orifice. It's a simple thing. Okay? This is a one type of organism, like. And then the another type, which is, I can just put it in a way like organisms like another one, where you are feeding, this is the mouth, and it passes through, and digests, and then some of them is absorbed in some places, and then finally it's being excreted out. And this is absorption which is taking place here. This is the animal is feeding. This is the pattern. I'm just giving an experimental, how this feed, and then it's digested. So one part is the mouth, and another one is the anus. One part into the other. So the digestion is a simple diagram. Some organisms, it comes out in one way, and this is the only one orifice. It comes and digests and will go out, right? Just come and absorption which is taking place once the digestion is over, and the waste product is being removed at the same orifice. The another pattern in the digestive system, the animal model, and another type of animals where the feed you have a proper mouth, and here the proper way out, but in between there's a digestive part. We are going to discuss about this part, how it differ here from different animals, how they differ here, then how they differ here, then what is the function of this and what is function of this, and what is the cellular architecture, and what are the hormones which regulate this movement, what are the enzymes which is involved in here in a digester. Here you have the complex food, but here everything is coming out the waste, but the ingredient, active ingredient of the food or the molecule is being absorbed. So our question or this topic is, we are at this level we are going to discuss on the digestive system. Okay? And also the membrane in how this is being moved from one to the other. That's again we call it as a membrane transport. Okay, membrane transport. Because whatever you eat, it should be across the membrane and then getting into the system. So how we are going to study that? To start with, there are several processes which involved. Okay, several processes which is involved. Food engulfed, okay, by membrane. We can call it as a membrane. Okay, food is engulfed by a membrane. Okay, and then digestion is taking place inside. Digestion intracellularly, and then the enzymes are in the vacuole. Enzyme vacuole. So this is a process, we call it as an endocytosis. 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 I put it SIS, endocytosis. I write it again, endocytosis, SIS, endocytosis. This is correct. Endocytosis. Okay. Food is engulfed by a membrane, like food is coming out, and this is being membrane is being engulfed like this. Okay, and this is one process. This is the F for food, and then the next process. What happened? This is going to be inside. Okay, this F is inside, and inside the cell. Okay, intracellularly is engulfed, and then you have vacuoles here, and this will digest this food material inside this cell. This is the one single cell, and thereby it is being digested, and then food particles is being absorbed and waste is being sent out. So this process, we call it endocytosis in a single organism, single cell organism. And for example, if you see, amoeba, have you ever heard of amoeba? It's organisms. So they engulf like this and then put it over there. They call it, the process, we call it as an endocytosis is a process. Okay. Another type of digestion, we call it as extracellular. Extracellular, where the food, okay, reduction of food extracellularly, reduction of food into small parts. For example, if you have a carbohydrate, complex carbohydrate, polysaccharide, these individual, all of them, yes for sugar. So they have a polymer of that. So if you take this polymer of starch, if you take the starch which is present in a potato starch, for example, if you have a fond of potato finger fries, French fries, if you eat that, if you have complex carbohydrates, okay. So if you take, you cannot swallow directly, but you are chewing them out, right? You are chewing them out. You are breaking down this one as a what? The amylase enzyme into starch, which will broken down into di or trisaccharides. So these are the broken down. They're simple. But they are extracellular. It is not taking place intracellular. This previous one is the endocytosis, which is an intracellular process. Digestion which is taking place intracellular. That the food coming directly onto the cell and then digestion inside the cell. But here, the food is outside the cell yet, but these are the enzyme secretions and then it's digestion, and then you go into the small, maybe complex, into the simple sugar molecules or the simple carbohydrates molecules. Okay. When you do that, and that reduced into the simple form, and then that is absorbed. So in this digestive process, we have to discuss about physics, how our organism or how any one of these organisms are using thermodynamic principle. Thermodynamics. Have you ever heard of thermodynamics before? Huh? Yes. Dynamics. Thermodynamics. 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 Okay. Just if you don't understand, this is not M, this is an N. Thermodynamics law. What is thermodynamics law 1? Law 1. Come on, if you know any physics, or you can understand physics now. Thermodynamics. They're going to thermodynamics. Yes, come on. Wonderful. What is your name? Good. Okay, energy. Conservation of energy. Conservation of energy. Energy conservation. So, first law says that energy can neither be created nor destroyed, right? Do you understand? So one form of energy is converted into the another form of energy. Energy is conserved. Energy is one entity. Energy cannot be converted from one. Energy can be converted from one form into the another form. Okay, you cannot destroy it totally. So energy is conserved. That's the law number 1, thermodynamics law. Do you remember that now? So very simple. Energy can neither be created nor be destroyed. Law 1. Very simple. Okay. Then thermodynamics law 2. What is that? Thermodynamics law 2. When the molecules from an orderly state to become a disorder. Very simple. I want to give. Or entropy of this universe is increasing, right? Entropy is increasing. So orderliness to disorder. So that is the law 2. So one example is the universe is becoming a second order thermodynamics principle, because the ever expanding of this universe is expanding. That's from one organization to the other. That's what the time of a Big Bang, they said, explosion of those. So it's going to be increasing, ever increasing entropy. In other words, when you keep it in the order, then according to the second law of thermodynamics, this became a disorderliness. Orderly to disorderliness. Keep in the order and then a disorderliness. Okay, entropy is increasing. If everybody is driving on a 70 miles per hour in the freeway, and all of a sudden a guy is going on a 10 miles per hour in the freeway, what is that one? What will happen? Yeah, it's a disorderliness, right? At the same time, if he is exceeding a 90 mile speed on that, that's also disorderliness. So it's an order to the disorder. So the entropy at that level, which is fluctuating, is increasing in entropy. So order to disorder. So when all the students are here at now 11:40 in this class, but at time 12:15, nobody is going to be here. Everybody is going their own way, taking their vehicle, their car, or lunch, and home, and everywhere. So what now it is order, it's become a disorder when you go out. Do you follow that? So the molecules are kept in the order. When you take your French fries in your mouth from your fast food, once you chew them up, what will happen? That orderly fries become a disorderliness. This is going to be increased. So that is the second order of thermodynamic principle. Do you follow? Everything is intact, but you are going to break it down, and then it's moving around. It's not going to be the same place. It's going to be moving around. It's going to be expanding everywhere. So that the principle which involves the second law of thermodynamics. That's a little bit of physics. You have to understand that. Okay. So what is the advantage? Why we have to study on the first order, second order here? We'll go through now. Okay. This one is low entropy, second order again. Low entropy, ENTROPY. Okay, low entropy to high entropy. Okay, that is orderly to disorderness. So why we used to study in digestion, we need some free energy. So delta G, so free energy, is equal to, I'm giving a simple equation, delta H minus T delta S. Okay, delta G is free energy. Changes, delta is changes, changes in free energy. That is equivalent to changes in H here, enthalpy or heat. Enthalpy, changes in enthalpy. Otherwise, we call it as a heat of reaction. That's what H for heat. Heat of reaction. Heat of reaction. Okay. Or we can also say probability of the reaction is taking place or not. So that's one other thing, enthalpy. And this one, delta S, we call it entropy, changes in entropy. Order, how far this order to disorder, entropy. And the T is the temperature, this one, T is temperature. Because the heat is produced here, heat of the reaction, that's on the temperature. So if you want to derive some free energy, that energy is the one we need for work to be done. Why we have to do the work? Why you have to study? Because physiology is nothing but how things work. You follow that? That's what the initial, when the start of the class, which I mentioned before. What is physiology? Physiology is nothing but how things work. If you want to work, you need energy. How that energy can be defined scientifically, that's what a free energy. The energy should be free. How you can get this energy? If you eat something, yes, you can get some energy. How that energy is be available in your system? Use enthalpy, heat of the reaction, and minus temperature, and then T delta S. The T times delta S. This is a change in entropy. How one molecule from orderly to disorderliness, that changes in orderly to disorderliness. That's one. And temperature. And then the enthalpy is the heat of the reaction, changes in the heat. This is minus, and this is the temperature of the reaction, T, constant, absolute temperature. And then you get changes in the free energy. The energy is none, and all on a sudden you have more energy because of this reaction. For this to be defined, you need to eat. If you don't eat, you get more energy. That energy, how we are going to get that process. So we are going to define these principles in terms of digestive system. Okay? We want to study the enzyme kinetics. As I mentioned before, this one, where the low entropy increasing and the low entropy to high entropy, how this process is taking place. We have in our system, or in the animal model, we have enzymes. Okay? The enzymes will do the work. We call it, in other words, enzyme kinetics. See, the enzymes which will do the job. Enzymes will do the job of what? Give the free energy. How this enzyme will work? Okay. And this will help, like a conservation of law number 1, right? Energy balance. Energy balance. That's the energy in, that's where you're eating it, that whatever we are eating it, energy input, and that will go into the system. Energy 1 plus 2 plus 3 plus 4. So the one entity of energy is going to be divided, or it's going to be, give rise to 1, 2, 3, 4 small part of this energy. For example, here 100, here the 25, and 25 is released, 25 amount of energy is released, and 25 energy is released. So if you think how much we are eating our food, and this energy is released in this form of 25, 25, 25. So energy input is equivalent to energy output. Otherwise, this is called a first law of thermodynamics. Okay, first law of thermodynamics: whatever we eat, and that the amount of energy that should be used in our system. Suppose whatever you eat more here, maybe you are eating 1,000, but you are spending the energy. How much you are spending? Maybe you are spending the energy 500. But what happened to the rest of the 500? That is the dangerous part, where you become unused energy, that become a toxic product, or that might be stored in our system. Hey, you ate too much, but I can work this much only. But your excess food, what? It cannot even spirit out of the system, but in turn, you switch on the other mechanism stored in the form of fat and accumulation of obesity, or the putting on the increasing the weight, or the increasing the glycogen storage, or the carbohydrates eating, or the fat that increases in your system, and that affects the disease and other process. Do you follow now? How the first law of thermodynamics in one place, and how that is going to be affected in your system. That's the one part. Second part of the law, or the second law, which comes on these enzyme kinetics. Okay. How the system will work? We put energy, okay, into the system. Okay. And now it works. And the part of the work should be liberated as heat. And then this work will be done as a waste product should be removed. Okay? The process of feces and urine. And then you also get sweat, which is coming out, and also breath, which is all the carbon dioxide which is coming out. So energy input here, energy in, that is equivalent to energy which is being spent. If it is not spent, what will happen? You get energy stored. So we want to avoid this one, but we want energy in, energy spent, this should be equal. Do you follow this part now? Any question on this one? A little bit hard. No? It's very simple. Did I explain everything in a simplified way? Yeah. If you don't understand, please, I can still find out something and then give some animation for that one and do that. Okay. Do you follow from Synchrorange? Yes? This, what is the first law of thermodynamics from Synchrorange? Jim? Sorry? Oh, okay, good, right. So what will happen if excess accumulates? Correct. Okay. Now, and I'll ask you another question. What is the second law of thermodynamics here in Sugarland? Anyone? Orderly to disorderliness. Okay, that's good. That's good. So what I'll do, see the first class, I'm not going to give that details onto you, but I'll go on to the next class. Again, the following is the digestion part from the second law of thermodynamics, and then the principles which is involved, and then following the digestion part. So I'll stop it here today, and then probably because time is up to 12:15, but I'm just giving maybe 15 minutes earlier. I will don't disperse the class, and hereafter when we discuss on this, I will prepare some questions, and once the class is over, and we will review through the question again, and I will ask you the question, you are answering the questions together.